Hello students, welcome to the class. Today we are going to discuss about chapter 9 Biomolecules. So much in detail regarding the biomolecules we will be learning in class 12, all the structural details as well as the functions, everything you will be dealing it in detail in class 12 chemistry. This is, as of biology is concerned, we should know some at least the basic concepts of these biomolecules, because the molecules are present inside the living organism. So it is a biomolecule which is present inside the living organism, that why the term bio is being introduced, isn't it? So here, moving on to these biomolecules in this chapter, for as of now we are just going to cover the bird's eye view of the chapter. Much in detail we are not going to go, we are just going to cover it up and we need to know the basic biomolecules which are present inside us, okay, a bit in detail but not much, right. Now moving on to this chapter, biomolecules. These biomolecules can be divided into two categories. So what are the two categories? The first category is biomicromolecules, means which are very small, okay. These biomicromolecules, when they combine together, they result in the formation of a bigger molecule which we call it as biomacromolecule. So two types we are going to discuss, one is biomicromolecule and another one is biomacromolecule. Other than that, we will also be discussing about enzymes, okay. So enzyme is also one concept which has to be discussed in case of this biomolecules. Now moving on to the first part regarding biomolecules, first we are going to see biomicromolecules. What we are going to see? We are going to see biomicromolecules. So how much I am dealing, that much is enough for studying as of now, okay. In detail in 12 you will be covering. Now moving on to this micromolecules. So when you are seeing these micromolecules, the weight of these molecules will be around 18 to 800 daltons. So some will be a bit heavier and others will be a bit light in nature, okay. So biomicromolecules are smaller units compared to the macro. The micro is smaller, isn't it? And when you are seeing, they are not that much heavy, they are a bit light, okay. Now this biomicromolecules can be divided into four major categories. So what are the four major categories? One is your sugar, second is lipids, third is amino acids, and fourth is heterocyclic compounds, okay. So sugar everyone knows, when you are moving into in our body there is lot of sugar, isn't it? This sugar is a form of carbohydrate, right. Now when you are seeing this sugar, when it comes to biomicromolecule, which means it is smaller, so it can be of two types. One is monosaccharide and another one is disaccharide. What do you mean by mono? Mono means if it is made up of only one sugar molecule, one glucose unit, okay. So then we call this one as mono. When it is made up of two sugar units or two glucose units, then we call them as what? We call them as disaccharides, okay. So you know that smaller things are combining together to make a bigger thing. Now we are going to discuss about what the smaller things. Like for example, when you see, we have learned the proteins, isn't it? So what is the smallest form of protein? It is amino acids. Now when it's amino acids, two amino acids combined together, they form what? They form dipeptide, isn't it? So like that, when many amino acids are combining together, they form polypeptide. So this polypeptide is bigger, amino acids are smaller. Like that, monosaccharide, disaccharides are smaller units, and polysaccharides like starch, glycogen are all what? Polysaccharides, which means they are bigger units. So biomicromolecules deals with all smaller units. So under sugar, what are the two smaller units of sugar which we are going to discuss? The monosaccharide, one is monosaccharide and another one is what? The disaccharide. For example, for monosaccharide it is given made up of only one sugar unit. What it is? Glucose, okay. Glucose is having only one sugar unit and it is the simplest form that can be easily observed, need not digest because already it is the simplest form, okay. Another one is your fructose, fruit sugar is an example for monosaccharide. Disaccharide, when you are seeing sucrose, you know what is sucrose? It is nothing but the sugar what we add, get it from sugar cane and what we are using in our kitchen, right? So that is sucrose. Sucrose, when you are seeing, they are made up of two sugar units. They can be broken down into monosaccharides, okay. And maltose is the malt sugar right? So this one also, when you are seeing this malt sugar is also made up of 2 sugar unit which can be broken down into monosaccharides, right? So this monosaccharide is the very simplest form, then it is the disaccharide, bit simpler form, okay. So these are the 2 types of sugar which is coming under biomicromolecules. Next we are moving on to lipids. What is the next biomicromolecule what we are going to discuss? It is lipids. Now when you are looking into this lipids are divided into 5 main categories. So what are the 5 main categories of lipids? If you see, the first one is fatty acids, second one is glycerol, third one is esters of fatty acids with glycerol, phospholipids and cholesterol. These are all lipids, types of lipids. What is lipids? Lipids is something related to fats, okay. They are fatty substances, means they are having some association with fats. So like oil or butter or ghee, dalda, whatever it is all we can categorize them under lipids, means something to deal with fats, okay. So this fatty acids, when you are seeing, there are 2 types of fatty acids. One is the saturated fatty acid and another one is what? Unsaturated fatty acids, okay. So saturated and unsaturated fatty acids are types of what? The fatty acids. Next fatty acid is your glycerol, yeah, one form of fatty acid, one form of lipid. Next is esters of fatty acids with glycerol, means they are having an ester group, you know, like ketone, structure is there, functional group, isn't it? Ester is one functional group, aldehyde is one functional group, like that esters of fatty acids are present. In that it can be categorized into 3 main categories. So what are they? One is monoglycerides, second one is diglycerides and third one is triglycerides. Monoglyceride contains how many glyceride unit? They contain only 1. Diglycerides is containing how many glycerides? Like monosaccharide contain 1 sugar, like that here monoglycerides will contain 1 glyceride molecule. Diglycerides will contain 2, tri will be containing 3. Now this triglycerides in turn is divided into 2 categories. What are they? Fats and oil. Fats, when you are seeing, what is the characteristic feature is that they have high melting point, okay, they need much amount of temperature, means they are having what? High melting point. For example, butter, right? And another one, oil, when you are seeing, they are having low melting point. Example is your gingerly oil which you use for putting in lamp and burning the same. So these are examples of what? Triglycerides. Next, phospholipids, example is lecithin, lipids with a phosphor group in it. And another one is your cholesterol which is present in your body is also coming under lipids. So these are the various types of lipids, biomicromolecule, which are divided into different types that we discuss. So fatty acids, saturated and unsaturated, glycerol, esters of fatty acids with glycerol, in that 3 are there. What are they? Mono, di and tri, isn't it? In this tri you are having 2, one is fats and oil, phospholipids, lipids with a phosphor group, and the other one is your cholesterol, are the different types of biomicromolecule called lipids, right? So next we are going to discuss about amino acids, okay. So we have discussed about 2 things already. So what were they? One is your sugar, second one is your lipids. Now we are moving on to what? Amino acids. So this amino acids, when you are seeing, there are different types of amino acids. So what are the different types of amino acids when you are seeing? One is your acidic amino acid, second is your basic amino acid, third is your neutral amino acid, and fourth is your aromatic amino acid. So what is the difference between these types? Basic, acidic, you know what is basic, what is acidic in nature, isn't it? So acidic, when you are saying they are having pH lesser than 7, and basic they are having more than 7, and the pH is 7 for neutral categories. So these are the different types of amino acids based on their pH, acidic, basic and neutral. Like glutamic acid is an acidic one, lysine is a basic amino acid, and valine, if you are seeing, it is a neutral amino acids. Another one type of amino acid is your aromatic amino acid. Aromatic Like for example, tryptophan and tyrosine is an aromatic. Aromatic, what do you mean by the term aroma? Aroma means they have smell, okay. So aromatic amino acids, when you are seeing, it is again a type of what? Amino acids. So these are the basic categories of amino acids. Now functional forms, when you are seeing this amino acids, in what form it can be present, okay. It can be present in 3 different forms. What are the 3 different forms? One is your fully protonated form, second one is your zwitterionic form, and the third one is your deprotonated form. What do you mean by protonated? Protons. Protonated, which means they have addition of a proton. H plus ion is added to the same. So then it becomes what? Then it becomes protonated. This is an example for. So amino acids have the functional group COOH. So what is the COOH? It is an acid, isn't it? So you know, OH is alcohol, CHO is aldehyde, COOH is acid, C double bond O is ketone, isn't it? So here when you are saying COOH is the amino acid, the functional group, R is referring to any one of the alkyl group. It can be CH3 or C2H5, like that. It can be of any alkyl group. And another one is your nitrogen containing group, amine group, okay, NH3. So these are the basic concepts, means basic structure. When you are saying they have a nitrogen containing amine group, acid group, and they are having what? A methyl group in it. Now when you are seeing this protonated amino acids, they have plus, they are having addition of proton, because of which they are positively charged. And deprotonated, as you know, the name suggests deprotonated, hydrogen is removed, so hydrogen is not there, so they are negatively charged. So when you are seeing into this amino acids, they are what? They are negatively charged, okay, and hydrogen is removed from them. Another one form is your zwitterionic form, means they exhibit both positive as well as negative at a time. So that type of ions we call it as zwitterionic form. You can find NH3 plus is also there, CO minus is also there. So both protonated as well as deprotonated form are equally exhibited in this form of amino acids. What is that form we call? We call them as zwitterionic form, okay. They are neither protonated nor deprotonated. They are not positive or negatively, means they are having both the charge within themselves. So that one we call it as what? The zwitterionic form. So these are the different types of amino acids, right? The 4 types we discussed: acidic, basic, neutral and aromatic. And when you are saying based on the functional forms, they are of 3 types. One is your protonated, that is having a proton in them, that is hydrogen ion plus. Zwitterionic, both the forms plus as well as minus are equally exhibited, and deprotonated, they are not having hydrogen, it's been removed, that's why they are minus in charge, okay. So this is regarding your amino acids. The next what we are going to discuss is heterocyclic compounds. So what do you mean by the term heterocyclic compounds? Listen carefully, it's a very important part, because you should have a base about the heterocyclic compounds, because in these things you are going to learn inheritance or as well as the DNA in detail in class 12, wherein these all terms are very, very important, okay. Now heterocyclic compounds are divided into 3 categories. So what are the 3 categories? One is your nitrogenous bases, second one is your nucleosides, third one is your nucleotides. Now listen carefully, dear. Now when you are seeing one biomolecule called as DNA, okay, it's a bigger one, macromolecule. So this DNA, when you are seeing, it is made up of nitrogenous bases. What they are made up of? Nitrogenous bases. So what the nitrogenous bases are? Okay, carbon containing group which are present in the ring, that one we call it as what? The nitrogenous bases. Now this nitrogenous bases are of 2 types. So what are the 2 types of nitrogenous bases? When you are seeing, they are purines and pyrimidines, okay. So purines are made up of 2, one is adenine and another one is guanine, okay. And pyrimides is of 3 types, one is cytosine, thymine and uracil are the 3 types of pyrimidine bases. Now this nitrogenous bases, I told you, it is divided into purines and pyrimidines. Purines are again divided into adenine and guanine. Pyrimidines are again divided into cytosine, thymine and uracil. Now listen, when you are looking into the structure of DNA, where what we are going to see, the structure of DNA, the DNA will be like this, isn't it? So in this DNA, if you are seeing here, actually the ladders, the DNA which the ladders it is present, there only you can find the nitrogenous bases are present. Okay. So if this strands, in one strand if it is adenine present, then opposite in the other strand. So in one strand if it is adenine present, opposite in the other strand what will be present? You can find thymine will be present. What will be present? Thymine, and within them there will be double bond. Okay. So adenine and thymine, if you are seeing, it is nitrogenous bases. Guanine, if it is present, it will be present with cytosine only. If cytosine is present, then it will be present with guanine. If thymine is present, it will be present with adenine. So from here what we can make out, what do we make out from here? We can make out that adenine always pairs with thymine. Adenine is always pairs with thymine. Guanine is always pairing with cytosine. Okay. So whenever A is there, opposite to it what should be there? T. And whenever T is there, opposite to that what should be? It should be A. The same, whenever G is there, opposite to it what should be there? C. And whenever C is there, opposite to it what should be there? G. So adenine always pairs with thymine with a double bond. Guanine always pairs with cytosine with a double bond. Thymine always pairs with adenine with a double bond. Cytosine always pairs with guanine with a triple bond. Okay. So between these two it is triple bond, and between these two it is double bond. Now then what is uracil? So adenine, guanine, cytosine, thymine and all is fine. Then what is uracil? See, listen dear. This, in case of RNA, this uracil is present, uracil is not present in DNA. In spite of thymine, you can find in RNAs, in the place of thymine what will be present? The uracil will be present, right. So like this you can find these are the nitrogenous bases in a DNA, the nitrogenous bases I told you, right. Now the next thing what we are going to discuss is that these nitrogenous bases, when you are seeing, they are attached to the sugar. With what they are attached to? They are attached to the sugar. Okay. So this, when they attach with the sugar, means nitrogenous bases with sugar, we call them as nucleoside. What do we call them? We call them as nucleoside, right. So if you are taking a DNA molecule, nitrogenous bases A, C, G like this, if it is present in one strand, other strand how it will be present? T, G, C, isn't it? So nitrogenous bases, when it is attached with sugar, what they are forming? They are forming nucleosides. So the nucleosides, when if it is adenine present with the sugar, adenosine, okay, and if it is guanine present, guanosine. If it is cytosine present, then we call it as cytidine. If it is thymidine present, then we call it as thymidine, and if it is uracil present, we call it as uridine. Okay. So these are nucleosides. Now listen very carefully. Another one thing which the DNA has is that, so I told you nitrogenous bases are present, the ladders, and it is attached to the strand with the help of sugar. Now this sugar is attached with phosphorus. With what it is attached? Phosphorus. Now this is sugar, isn't it? In between what is present? P is present. So when I am drawing, how does the strand becomes now? Phosphorus is also being added. Now the DNA will become complete. Along with the in between proteins will be there. But as such DNA, if you are saying sugar, phosphorus and nitrogenous bases. Okay. So nitrogenous bases, when you are seeing how, when I am drawing a strand, how it was, it is like this, isn't it? So this is a strand. So if adenine is present, correspondingly there will be thymine. Now this adenine is added with sugar. Sugar, then it becomes what? Nucleoside. When it is adding with phosphorus, it becomes what? It becomes nucleotides, right? So if you are writing it just like that clearly, so how it becomes? A double bond T, C triple bond G, G triple bond C, T double bond A. Now here it is attached to sugar, sugar is attached with phosphate, again phosphate, again sugar, like this. You can find phosphate, then again sugar, like this the strand will be present. Now when this sugar, nitrogenous bases and phosphorus are adding together, we call them as nucleotides. nucleotides. What do we call them? We call them as nucleotides. Okay. So, heterocyclic compounds, I told you, DNA or RNA is the biomacromolecule. So, when you are moving on to that detail, when you are seeing this DNA or RNA, bigger molecule are made up of smaller molecules, the heterocyclic compounds. It's of 3 types. So, what are they? The smallest one is the nitrogenous bases. They are of 2 types, purine and pyrimidine. Purine is of 2 types, adenine and guanine. Pyrimidine is of 3 types, cytosine, thymine and uracil. Now, nucleoside, when you are seeing, when these nitrogenous bases combine with sugar, they form nucleoside. And when this nucleoside, that is the nitrogenous bases with sugar, combines with phosphorus, then it becomes what? Phosphoric acid or phosphate. They becomes what? They becomes nucleotides. Adenine, if it is forming, means adenosine, if it is combining with phosphorus, it becomes adenylate acid. Okay, guanylic acid, cytidyllic acid, thymidyllic acid and uridylic acid. So, these are all nucleotides. How is nucleotide formed? Nitrogenous bases plus sugar plus phosphorus. Only nitrogenous bases and sugar is what? It is nucleoside. What it is? Nucleoside. When all are present, we call it as what? We call them as nucleotides. So, with this I am stopping for today. Hope it's clear to every one of you. Thank you.